Bismillahirrahmanirrahim. In the name of Allah, the most beneficent and merciful. Student, Assalamu alaikum. My name is Dr. Rana Farhat Mehmood, and today we are going to discuss our third chapter about X-ray diffraction technique under the course code of Chem 5134. And this third chapter is about single crystal X-ray diffraction. In this slide, we are going to show you some animation about single crystal X-ray diffraction, in which we have some X-rays coming out from the X-ray tubes, and it is passing through the monochromator, and after passing through the monochromator it strikes on a single crystal in their path, and after striking it is scattered on certain angles, and then it is recorded on the film or recorder at the end of this animation, and there it gives different pattern relating to the atoms attached in a single crystals, which we can get different information about the structure of the single crystal. Uh, Here is a single crystal X-ray diffraction instrument which is used to determine the structure of the single crystal, and in this machine there is a X-ray tube and a goniometer on which the single crystal is mounted, and a detector which records the scattered beam after striking the single crystal. So here in this slide we have a brief introduction about the single crystal X-ray diffraction. In the introduction we can see that in an X-ray diffraction measurement, a single crystal is mounted on a goniometer and gradually recorded while being bombarded with X-rays, producing a diffraction pattern of regularly spaced spots known as reflection. First of all, the two-dimensional images taken at different rotations are converted into a three-dimensional model of the density of electron with the crystal using mathematical method for Fourier transform, combined with chemical data known for the sample. Poor resolution or fuzziness or even errors may result if the crystals are too small or not uniform enough in their internal makeup. So here we can see that in X-ray diffraction machine, a single crystal is mounted on a goniometer, which is rotated all the time when the X-rays are being bombarded on the crystal. That produces a diffraction pattern of regularly spaced spots, and these diffraction patterns are known as reflections. First of all, the two-dimensional images are taken at different rotations, and then these two-dimensional images are converted into three-dimensional model of density of electron, and this is converted with the help of mathematical method of Fourier transform, which is also combined with the chemical data known for that sample. And if we have a crystal which is in very small size and it is not uniform enough in its internal makeup, then the resolution becomes very poor or fuzziness, and this also create some errors in the result. So the crystal size should be large enough to get the good result. And if the single crystal of the sufficient size cannot be obtained, then various other X-ray methods can be applied to obtain less detailed information. Such methods includes fiber diffraction, powder diffraction, small angle X-ray scattering. That means if the crystal is not in a large size or in sufficient size, then we use different other X-ray methods to get the detailed information about that crystal, and these methods are fiber diffraction, powder diffraction, and small angle X-ray scattering. And if the material under observation is only available in the form of nanocrystalline powder, which is also a very small in size, or it suffers from poor crystallinity, the method of electron crystallography can be applied for determining the atomic structure. So, for nanocrystalline powder size crystals, the method that we use is electron crystallography, and it is used to determine the atomic structure. And for all the above mentioned X-ray diffraction methods like fiber diffraction, powder diffraction, small angle X-ray scattering, the scattering is elastic. The scattering is elastic mean the scattered X-ray have the same wavelength as the incoming X-rays. That means no change in wavelength occurs when the X-rays strike the single crystal and it gets scattered. So both the incident X-rays and the scattered X-rays have same wavelength. And by contrast, inelastic X-ray scattering occurs, means if the change in wavelength occurs between the incident X-rays and the scattering X-rays, then other methods are also used to study the single crystal X-ray diffraction. And these methods are useful in studying excitation of the sample rather than the distribution of its atoms. The oldest and the most precise method of X-ray crystallography is single X-ray diffraction, in which a beam of X-ray strikes a single crystal and produces scattered beams. When they land, means when these beams are struck on a piece of film or other detectors, these beams make a diffraction pattern of spots. The strength and angle of these beams are recorded as a crystal as the crystal is gradually rotated, and each spot is called a reflection, since it corresponds to the reflection of X-rays from one set of evenly spaced planes within the crystal. Dear student, in this slide, we are going to tell you that if single crystal is of sufficient purity and regularity, then X-ray diffraction data can determine the mean chemical bond lengths and angles. And these chemical bond lengths are between the atoms. That is, the distance between the atoms can be measured by X-ray diffraction data. And also the angles between the atoms or the planes of the crystal lattice can be measured by X-ray diffraction techniques. And also we know that the atoms in a crystals are not static, but oscillate about their mean position, usually by less than a few tenths of an angstrom. So, by using X-ray diffraction technique, X-ray crystallography allows the measuring of the size of these oscillation. It means by using the X-ray diffraction techniques, we can easily measure the bond length, bond angles, and we can also measure the size of oscillations of these atoms in a single crystal of sufficient purity and regularity. Why we take a single crystal and why we use X-ray diffraction technique? By the way, that means why is a single crystal necessary and why are X-rays used for X-ray crystallography? It is used for viewing the three-dimensional structure of molecule quantitatively. It is required to satisfy the following three conditions. Number one, type of atoms are distinguishables. Number two, the position of the individual atom which is buried inside the molecule can be determined even the molecule is large like protein. And number three, the view of molecules from as many different directions as possible can be obtained. In addition, the relative position among the molecules from different views can be determined precisely. So these are the reasons why we use X-ray crystallography. It is used to view or to determine the three-dimensional structure of the molecule quantitatively, and to determine the structure quantitatively in three dimension, it requires three conditions. One is type of atoms must be distinguishables. Number two, the position of the individual atom in which it is buried inside the molecule can be determined even if the molecule is large like protein. Number three, it is also used to view the molecule from as many different directions as possible and can be obtained. In the next slide, we there is a animation of a single crystal, and in this it is the X-ray crystallography that satisfies these conditions. Then currently it is most popular analytical method to solve the three-dimensional structure of the molecules. The third condition is certainly satisfied by the nature of crystals. So we have different type of crystals, and in these crystals the length between the atoms and the angle between the atoms are different, which divide them in different categories. Once again we discuss that why XRD is used. So it is used to measure the average spacing between the layers or rows of atoms. XRD is also used to determine the orientation of a single crystal or grain. XRD is also used to find the crystal structure of unknown material. It can measure the size, shape, and internal stress of small crystalline regions. So these are the main aspect of XRD that can be used to measure the average spacing between the layers or rows of atoms. It is also used to measure the orientation of single crystal. It is used to determine the structure of unknown material, and it can measure the size, shape, and internal stress of small crystalline regions. Here is a comparison between single crystal X-ray analysis and other methods. So recently, elucidation of molecular structure is becoming more common owing to the development of various measurement techniques. And there is a following table data in the next slide. But when we see that nuclear magnetic resonance, NMR, mass spectrometry, MS, and infrared spectroscopy, IR, are the typical examples which are used to elucidate of molecular structure. And these techniques are also being used relatively with respect to X-ray analysis. However, these spectroscopic techniques derive just a little of partial structure, just a list of partial structures, and it is sometimes difficult to deduce the three-dimensional structure of whole molecule. On the other hand, when we use X-ray diffraction technique, it is the molecular structure itself that is derived from the single crystal X-ray analysis. The single crystal analysis provides a unanimous conclusion that sometimes puts an end to argument over molecular or crystal structure. Here is a chart that have some comparison between the different techniques single crystal XRD, NMR, MS, and IR, and how we can determine the properties of a crystal by using these techniques. These are all discussed here relatively, and which one of them is a non-destructive measurement technique, and for which technique we have to prepare sample, and sample should be in the form of crystal or liquid solid, solid liquid gas, solid liquid gas. Samples can be used in different techniques, and measurable atomic types. So SCRD can measure the all type of atomic sizes, and there is other properties like determination of configuration, determination of absolute structures, and determination of crystal structures, sample preparations, and data interpretation. And here we can see that single crystal XRD technique is superior to all other technique in determining the structure of the atom. Fundamental principle of single crystal X-ray diffraction. Here we have a look on that how fundamental principle of single X-ray crystal diffraction are invented. Max von Loo in 1912 discovered the crystalline substance act as three-dimensional diffraction grating for X-ray wavelengths, similar to the spacing of the planes in the single crystal lattice. X-ray diffraction is now a common technique for the study of crystal structure and atomic spacing. X-ray diffraction is based on constructive interference of monochromatic X-ray and crystalline and a crystalline sample. So these are the fundamental principles that were discovered by Max von Loo in 1912. And in the next slide, we see There are some other fundamental values. These X-rays are generated by cathode ray tube, filtered to produce monochromatic radiation, collimated to concentrate, and directed towards the sample. The interaction of the incident rays with the sample produce constructive interference and a diffracted rays. When conditions satisfy Bragg's law, that is n lambda is equal to 2d sine theta. This law relates the wavelength of electromagnetic radiation to the diffraction angle, at which angle they are scattered after striking the single crystal, and the lattice spacing in the crystalline sample. The Bragg law gives the relationship between the wavelength coming from the X-rays and the diffraction angles which occurs after striking the single crystal, and it also gives information between the spacing of the two layers of a crystalline material. These diffracted X-rays are then detected, processed, and counted. All diffraction methods are based on generation of X-rays in the X-ray tube. Here is a source. Here is animation which is showing that X-ray source. X-ray source is the X-ray tubes from where X-rays are coming, and this red line is X-ray beam which is striking on a single crystal, and after striking the single crystal it is scattered to different angles and gives different mode of scattering. And because we know that this crystal is always in a state of rotation, and it gives a different kind of diffracted beams which can be recorded in a detector or on a film. By changing the geometry of the incident rays, the orientation of the centered crystal and detector, all possible diffraction direction on the lattice should be attained. These X-rays are directed to the sample and the diffracted rays are collected. A key component of all diffraction is the angle between the incident and the diffracted rays. Powder and single crystal diffraction vary in instrumentation beyond this. Dear student, now we are going to discuss some importance of the single crystal X-ray diffraction technique. We know that single crystal X-ray diffraction is a non-destructive analytical technique which provides detailed information about the internal lattice of crystalline substances. Non-destructive technique means the sample we use to analyze in this technique is not destructed by this technique, and this include unit cell dimensions, bond length, bond angle, and details of the site ordering. That means it is the most important technique in which the sample is also not destroyed, and it also provides detailed information about internal lattice of crystalline substance. It provides information about unit cell dimensions, bond length, bond angles, and also give information about the site ordering. Directly related to the single crystal refinement, where the data generated from the X-ray analysis is interpreted and refined to obtain the crystal structure. That means The data that is generated from the X-ray analysis is directly used to uh, relate the single crystal refinement, and also it is used to obtain the detailed information about the crystal structure. In explanation, we see that the analysis of a diffraction data obtained from single crystal is the most important method of obtaining the structure of inorganic solids. As we know that inorganic solids are the solids which are found in the crystalline state. So these inorganic solids and the crystal of these inorganic solid are studied by using single crystal X-ray diffraction technique. And this provides and it is provided a compound can be grown as a crystal of sufficient size and quality. The data provide definitive information about molecular and extended lattice structure. That means for this information that we collect, definitive information that we collect from the crystal, for this we have to grow a crystal of sufficient size and quality. And the collection of the diffraction data from a single crystal is normally carried out by using a four circle or area detector diffractometer, which is shown in the coming slide. A four circle diffractometer uses a scintillation detector to measure the diffracted X-ray beam intensity as a function of the angles shown in the illustration. Here is the illustration of the four circle diffractometer. So in this figure, the layout of four circle diffractometer is depicted. A computer controls the location of the detectors as the four angles are changed systematically. Here we can see that there are different four circles in which sample is present, X-ray beam is also used, and a detector is used. And this detector is used to detect the scattered beam after striking the single crystal. Here is another layout of the image and of the process that take place in a X-ray single crystal diffraction technique. The layout of four circle diffractometer, in which it is also told that computer control the location of the detector as four angle are changed systematically. An area detector diffractometer uses an image plate that is sensitive to X-rays and so can be measure a large number of diffraction maxima simultaneously. Many new systems use this technology because the data can be typically collected in just a few hours. Analysis of the diffraction data from single crystal is formally a complex process involving the location and intensities of many thousands of reflections. But with increasing the advances in computation power, a skilled crystallographer can compete the structure determination of a small inorganic molecule in under an hour. Single crystal X-ray diffraction can be used to determine the structure of a vast majority of inorganic compounds with when they can be obtained as crystal with dimensions of about 50 into 50 into 50 micrometer or larger. Thank you very much, dear student. We will come back in the next lecture with the remaining information about single crystal X-ray diffraction technique and its importance and its practice. How can we use this technique to determine the single crystal of the inorganic solid or any crystalline material. Thank you. Allah Hafiz.